is Aidan Riley from Boston University, PhD candidate in biomedical engineering.
All right, hello everybody. I was told I look nervous before this, and it's because I am, but here we go. So my name's Aidan. I'm a PhD candidate in the Green Lab at Boston University, where we do computational design of different synthetic biosystems with RNA as our molecule of choice. And recently what we've been working on is two complementary frameworks for predicting the function of RNA molecules and designing new instances of those molecules using generative adversarial networks. And so if you are familiar with RNA experimental biology, you will know that the high throughput screen is sort of the bread and butter experimental technique for trying to map the sequence to function relationship, regardless of the class of RNA. What we've noticed, though, is that a lot of the models that are published alongside these data sets only accept a one-hot encoded sequence input. But as RNA biologists, we know that secondary structure is one of the most impactful attributes of an RNA molecule to describe how it functions. So what we did was develop a generalized architecture that accepts paired one-hot encoding and structural array representations of RNA molecules into a lightweight predictive architecture that can be rapidly retrained on diverse classes. So you can see on the y-axis here of this nicely colored plot, we basically retrained this on essentially every high throughput data set that we can find so far, from minimum free energy to different types of guide RNAs, untranslated regions. So all these things that have sort of unique functions that could be really interesting for synbio purposes, we want to be able to predict in a very rapid manner. We've also found that using these structural arrays can drastically reduce the number of hyperparameters that are required to match or improve upon the state-of-the-art models. This is really important because if you want to send these models out to your experimental colleagues, you want them to be able to run on personal compute hardware. And then essentially the question becomes, if you have a nice predictor that you can apply in these different settings, how do you generate using this predictor to inform your process? And I'm sure many of you are aware of generative adversarial networks. These are a really cool technique that have the advantage of being plug and play with the predictive models. So what you can do is you can train a generator to design a specific class. You can plug it in with the predictive model that operates on that same class, and you can push your generated sequences towards a region of high experimental activity. And so quickly, the design task that I wanted to share with you guys is the toehold switch. So this is a riboregulator that we developed in the lab. My advisor did when he was a postdoc. These molecules essentially control the expression of a protein in response to a programmable trigger. And these triggers have no sequence constraints at all. They can hypothetically be any 30 nucleotide sequence. And these stems here will actually hide the necessary sequence motifs from the translational machinery unless the trigger RNA is there to unwind the stem and expose the ribosome binding site and allow translational readout. So this is a really good example for the utility of these models, because here we have a combination of sequence motifs that need to be included, structural characteristics, and very importantly as well, a lot of optimization techniques sometimes will land on a single best sequence, but these molecules need to be programmable to any target RNA, which is a very important design constraint that we see carrying over to new RNA systems in the future. And as you can see down here in the corner, we have tested experimentally the sequences returned by our generative program against the most widely used structurally informed inverse design tool. And you can see we get a nice increase of about 43x when we calculate the on-off ratios of these sequences in E. coli. And yeah, that concludes my talk. Thank you very much.
For the RNA generator, I was curious about the, so you were utilizing these sequence and structural embeddings. Have you thought about using more higher dimensional representations of RNA, like the RNA transformers that have recently come out, to see if that would improve the predictive capability of your model? Or are you going to stick with these more lower dimensional embeddings that you're using, like one hot and the structural encodings?
So that's a really good question. I think it's definitely a really good idea. We have sort of an experimental focus, I would say, in our group. So I think our long-term interest is get a model that works well and then see what you can do experimentally with those sequences that you design, right? So I think there's probably a lot of different embeddings for RNA structure that will continue to improve. But yeah, the emphasis on simplicity of the models and getting to the experiments are big for us. So thank you.